So thank you for joining us for this evening's BMS Talk. It's my great pleasure to introduce our chair for this evening, Ali Ashby. Ali is a fungal biologist and science communicator, formerly a Royal Society University Research Fellow and member of the Department of Plant Sciences at the University of Cambridge. Ali is also a long-standing member of the British Mycological Society. So thank you very much, Ali, and over to you.
Thanks very much, Sally, and welcome everyone to the final session, I believe, of BMS Talks for this year. So this evening, we're going to explore fungi that affect our food and our health. And I would like to welcome our two speakers, Anastasia Sokolidi and Barsha Kalita, who will be presenting talks based on their PhD research projects. Each talk will last about 25 minutes, and I would ask that the audience post any questions for the two speakers into the chat during their talk. And at the end of their talk, I will then bring those questions to the speakers. So our first speaker this evening will be Anastasia.
Hi.
Okay, and Anastasia holds an undergraduate degree in biochemistry from the University of Edinburgh and a master's degree in crop improvement from the University of Nottingham. Her PhD focused on aerobiology and the development of molecular diagnostic tools for the horticultural industry at Rothamsted Research. She's currently working at ADAS as a crop pathology consultant, where she will focus more on the applications of research within the agricultural field and with a particular interest in early disease detection methods. So this evening, her talk is entitled Early Detection of Airborne Fungi for Improved Disease Management in Commercial UK Glasshouses. So it's now up to me to hand over to Anastasia to present her talk.
Thank you very much. Bear with me whilst I share my screen. Ali, is that playing all right?
Yes, that looks good. Thank you.
Fantastic. Thank you very much for the introduction. I'm here to talk about, indeed, the early detection of airborne fungi for improved disease management in commercial UK glasshouses. Now, there's different methods and ways in which we can approach this topic. However, I have approached it just one of the ways through the use of spore traps. Now, spore traps are, in essence, hoovers that have been left with scientists for a very, very long time. So we have adjusted them to sample pollens and fungal spores in the air at a constant rate, which we trap in little test tubes or on sticky tape, which we then analyze and process under the microscope. In essence, the reason why we're doing it is because it allows us to predict how much plant pathogenic fungi is in the air, at what concentrations, and allows us to detect it even sometimes before it infects the plant itself, and we can see visible symptoms, thus allowing us as much time as possible to predict how the disease is going, when the disease is going to start, and when sprays need to be applied. This has been very, very useful in fields. In this case, you see some spore traps in an oilseed rape field. I think a particular couple of cases that I wanted to mention was when people measured for, in the US, for powdery mildew in grapevines, where they found that by the use of spore traps and the analysis of the tapes and the samples, they have managed to emit 2 sprays early on in the season, saving growers approximately 5K every single season when sampling and using spore traps. Another way to deploy it is to use this technology over the full season. In this case, it was used, an example of this would be for potato blight. where expensive fungicides were only applied when there was a high influx of spores in plant of potato blight. So they could time stronger fungicides with that to protect their crop. However, one of the main setbacks of spore traps a lot of the times is the fact that in order to analyze the data, we need to use and combine it with people and specialists in the field who need to sit there and spend a lot of time looking at the tapes that we produce through microscopy and to be able to identify what they see, what the species are. Now, this is very, very time consuming, and this eats into our time between which we can collect the sample and report the spore concentrations and the presence of pathogenic fungi to the growers, which would actually use this disease. So in order to omit that, we have started coupling spore trapping technology with molecular diagnostic tools. Now, there are many molecular diagnostic tools, but within my PhD, I have focused on molecular diagnostic tools that target DNA. And by that, there's a couple of methods that we generally use. They're called PCRs, qPCRs, and LAMP assays. In essence, what we do is we target a very, very specific small segment of DNA within a fungus of interest. And what we do is we design our assay to just focus on that small target, mix it with reagents when we get our sample, get the DNA out, mix it with the reagents, then put it through a cycle of heat treatments to amplify that very, very small target. And the way we measure it in our case is that we monitor this amplification of this DNA fragment over a set of cycles or a span of seconds and a span of time. What we're looking for is the increase of fluorescence that this technique does with the increase of DNA within the sample, which happens as time goes on. And the faster that this fluorescent peak increases means that we have more DNA than if it increased later on. And that is what we're looking for. Now, this technology can be used for different methods, but when we're coupling it with spore traps, we're looking at a specific fungus. And because our sample comes from the environment where there's a lot of fungi, a lot of pollen mixed in, we want it to be exceptionally specific to our fungi of interest. Then we also want it to be very, very sensitive, because a lot of the times spores of fungi only contain 1 picogram, which is quite a lot less and tiny. And a lot of our assays don't necessarily go so low, but we need it to be very, very sensitive because it allows us to detect the spores earlier. And because we do have an environmental sample that we're working with, we usually want to design a molecular assay that's more or less tolerant to inhibitors. And that is our goal. So during my PhD, we have developed one of them, particularly targeting one of the pathogenic fungi in the glass houses. Now, I have explained to you before that this system works very, very well in the field and has shown really good signs where it is applied. But our question was, will this actually work in a commercial glass house? Since we didn't know, obviously what we did is we pretty much went to some tomato growers and said, would you let us put in some spore samples, spore traps in your glass house? And they said yes. Now, these are some photos of how a commercial glass house looks. They're not your average standard glass houses. They tend to span at least a hectare. So sometimes you don't see the end. And when you put a spore trap within it, as you can see on the right in the picture, it looks very, very small compared to the size. However, one of the things that we wanted to do was to target it to a very, very specific fungus. So let me introduce the love and bane of my life. This is tomato powdery mildew. It is one of the main fungi that growers spray in commercial tomato glasshouses in the UK. It starts off very, very small and quickly goes to colonize the whole leaf. It's a biotrophic fungus, and it competes with our tomato plant for its nutrients. This reduces the quality of the crop, and in very severe cases can cause to plant death. It mainly grows over the tomato leaf, and what causes its powdery appearance is the conidiophores that grow out perpendicular to its matrix, which covers the leaf. And on top of it, you can see on the right, the little circles that form are the spores that get spread with mechanical disturbance or through air. And they get spread passively. And those are the ones that we will see in our spore traps when we're trying to trap them. However, there wasn't a lot of research to tell us about how exactly tomato powdery mildew spreads through the glasshouse. So we decided to just look and see what happens and monitor it. There's a couple of trends that we pointed out, and they're visible here in these 3 bubble plot diagrams of glasshouses, where the red circle you see is tomato powdery mildew, and the bigger the circle, the more the disease intensity at that particular point. The line down the middle of those glass houses represents a main pathway that usually workers use to travel down and with machinery and other items to get to the rows through which they just pass. And one of the things that we observe is that tomato powdery mildew is actually mainly spread along that main pathway, and that's usually its starting points. It's more likely to spread later on down along the rows than across the rows. And when we explored this topic a little bit further through a bit of modeling and some variograms, we realized that in space, when we see powdery mildew, we will usually see another point of powdery mildew within 25 to 50 meters from the source, without it having to infect the whole glass house. So, armed with this knowledge, does it actually matter where we put a spore trap within that glass house now that we know where the disease occurs? So we tried to address that by putting 2 different spore traps within the glass house at different distances and locations. And we tried to see whether the distance from our source of powdery mildew will affect how easily we will trap it. And obviously, we see that for both the different spore traps, the closer the spore trap is to the source of inoculum, the higher the spore count. And then as we go along, spore counts decrease, with us being able to securely detect, I would say, from around 25 to 40 meters away from the source, which means probably that we'll need quite a lot of spore traps if we just put them willy-nilly along the glass house. So it's probably better to put it in strategic areas, such as where disease primarily starts, so along our main pathways. Now, then we come to the main reason of why we're doing it. Can spore traps actually detect the presence of tomato powdery mildew before our growers? And can that be informative enough for them to then adjust their spraying strategies and policies to tackle the disease early on and only when necessary? Now, in orange here, you will see spore concentrations, whilst in the bars in dark blue or blue is actually the disease score on average within the glass house. Now, overall, these are two graphs from the same glass house over 2 different years. And we see that in both cases, the spore traps actually managed to find and have a peak of spores before I have counted and recorded it within the glass house. We also see that peaks correlate with disease intensity, which is good. Now, unfortunately, we have only managed to correlate this data with actually data received from our growers only in year 2022. And the moment that the growers actually recorded the presence of tomato powdery mildew in the glass house is represented by that black bracket in the year 2022 graph. And by that point, our spore traps already did detect the presence of disease, and we already knew that tomato powdery mildew would have been there. So it works to a certain degree, and hopefully it can be informative. However, we did also spot some problems within this method because when we put spore traps in a glass house within the same year that has a slightly different structure, we noticed that the peaks of spores were not as intense. And even though they do appear earlier, we couldn't really correlate it with the intensity of disease that we saw in it, even though we saw that its constant presence and the fact that spores were being there and emitted. Now, a lot of this must have been affected by the airflow through the different glasshouse and the different glasshouse structures. And there's a lot more to explore as to why this is happening and how reliable spore trapping can be within commercial glass houses to let our growers know when tomato powdery mildew is there or not and how useful it can be to them. But overall, I think the conclusions of my little PhD project have been quite positive. And on that, I would like to conclude that and go over the main points brought today, because we did manage to see that tomato powdery mildew mainly spreads along the main pathways. So we assume it's distributed by agricultural workers rather than air, and mainly machine flow that potentially causes the air. That tomato powdery mildew is usually present within is correlated within 25 to 50 meters in space. And that's where you should be looking for disease. And spore traps only detect powdery mildew approximately 25 to 40 meters away from the source of powdery mildew. But they can still detect, when placed strategically, tomato powdery mildew before growers, and are usually correlated to when concentrations and peaks of disease actually occur. And as such, to conclude, I would like to acknowledge all the team that has worked with me on it, my lovely supervisors and all the people at Rothamsted, particularly Dick Webster and Suzanne Clark, who helped me with my models and my stats and design. And I would like to highlight the members of the APS who actually were there working in the glass houses and helped me set everything up, as well as my funding bodies. Thank you very much.
Thank you very much, Anastasia. That was really fascinating. And clearly, you were very successful in achieving lots there in your PhD. I'm just going to check to see if we have any questions. Okay, let's start. We've got a question here, I think from Faye Ritchie. What would you recommend to prevent the spread of tomato powdery mildew in glass houses?
Oh, That is a very difficult question, actually, because there's many different ways in which we could tackle it. Usually it would have the main way that we reduce the spread is by actually fungicidal spray and applications. I would be cautious to give out any recommendations as yet, but that is the main strategies. All I can recommend is that we only apply sprays when we actually see the disease, which is something that doesn't usually happen in traditional commercial glass houses, because sprays are applied prophylactically. And a lot of the times they're not needed and only needed when the disease actually occurs and we see it in the glass house.
Great, thank you for that. So then we have another question. Thank you for your interesting talk. Should we be using microbiological protocol for staff entering glass houses and even microfiltration of air feed?
Ooh, I think we should. I think anything we can do to help us prevent the entry of fungi and parasitic fungi would be beneficial. And if that is something that we can do to intervene, I see no reason why we shouldn't be focusing on that and investing time in it.
Yeah, because presumably you're saying that most of the infection is coming from workers and machinery. So I'm guessing if you were able to, I don't know, disinfect down or something before you go in, or have some kind of sort of airlock system where you could walk through and clean up, go in, or
I think they definitely once the powdery mildew is there, they're definitely spreading it and they're helping it spread. I don't think it would be able to spread without them, because usually airflow within a glass house is quite low. So it wouldn't be able to spread that far. And the fact that we see it spreading so wide and far, they would have to assist with it. It travels on clothes, it travels on everywhere. I don't think it necessarily means that they are bringing it in, but it does come through mainly pathways, because that's probably where airflow is largest and has more chances just in terms of movement to actually go and infect.
And how quickly does the infection take hold once present in the greenhouse environment?
Well, when the spore lands on the leaf, it germinates within a couple of days. And I think for it to start producing spores, it takes a whole 10 days. And that would already mean that it's started to spread. And once it starts doing that, it will continuously keep spreading unless it is stopped. So.
Great. So another question from Cam. I presume that fungal spores of other parasitic species would travel through a greenhouse in the same fashion?
Yes, I think it depends a lot on the way that they're also produced. So powdery mildew has a very passive way of spreading, as it waits for the wind. I mean, other species tend to spread in more active ways. So it really depends on the fungus. However, if it's passive, yes, it will tend to spread similarly through the glass house.
And you were saying about you positioned the spore traps very carefully, obviously along the lines where workers were moving to and from. Is there? I mean, do you need to position them at any particular height? Did you notice for your work, or?
That's a good question. I decided not to include a slide on this, but we also did run an experiment where we have positioned spore traps at different heights, above, in between, and below the canopy, to see if our concentrations would be higher at any one point or any one day. However, we didn't see much of a difference at the points. So there was no significant difference between spore concentrations at the top, in the middle, or at the bottom.
So you opted for the middle, which is
so we opted for where we think it enters after modeling it and understanding how it spreads.
Linda Williamson asks, why not remove infected leaves and burn?
Ooh, I actually asked myself that question quite a lot when I was there. And I think this is more directed to one of the growers in terms of practicality. Because one of the things they already do within commercial glasshouse, one of the things they do is they deleaf the bottom leaves and they drop the plant because it keeps growing throughout the whole season, throughout the whole entire year. So technically they are removing the foliage and dropping it down. And they would be removing disease. However, at certain times, if powdery mildew starts at the top of the plant, you just wouldn't be able to remove that much foliage from a hectare, just from practicality standpoint. And then if you do, you end up losing a lot more photosynthetic tissue, which will almost be more of an impact. And the more leaves you remove, you also create more wounds, which allows other fungi, for example, to infect.
Another question we have from Akinwunmi Latundi Dada. I hope I've got that right. Asks, Fulvia fulva or Cladosporium fulvum? Question mark.
Come again?
I think they're asking what the powdery mildew pathogen is, I would think, which is I would think, rather than...
Oh, it's Pseudoidium neolycopersicae.
Yeah.
So we have two species in the UK. One of them is Liviula tarica. One of them is Pseudoidium neolycopersicae. However, we only spotted one of them within commercial glass houses and not the other one.
Right.
So we don't know where that one went.
And I mean, do they infect differently? I mean, how different are they in terms of their ability to infect in the glasshouse environment?
Oh, I wouldn't know the answer to that, I'll be honest with you. They do share some similarities, but they're quite different.
I'm just wondering whether you might see one more commonly sort of outside rather than inside, possibly. But...
Oh, I don't want to say anything wrong.
No, that's fine.
So
just intrigued.
It's just that I don't remember as much about Liviula tarica, because after we didn't detect it in commercial glass houses, I didn't.
I mean, it may have alternate hosts as well, I suppose, possibly.
They're quite broad spectrum, yes.
Yeah. So another couple of questions here. Are glass houses being deep cleaned between crops?
Yes.
Yeah. I guess that's standard practice, isn't it?
It is standard practice.
Yeah. And another question here: does UV light have any potential for controlling of mildew?
Oh, that is an exciting question. I think it has been very effective in strawberries for controlling powdery mildew. I don't know about tomatoes yet. I think there's mixed opinions on that one.
And the other question I have is, are there any other techniques that can be used, like sort of electrostatic techniques? I mean, I've come across things in the past where they can actually kind of suck spores off the leaf.
Yeah, that's one of the
powerful,
methods that we can deploy.
Electrostatic, yeah. Would that work, do you think, in a tomato glass house? Or is that crazy?
I think with no. It's not crazy at all. I mean, spores have a charge to them.
Yeah.
And there are spore traps that do trap spores electrostatically. I think
it's like an electrostatic
vacuum, the problem,
isn't it? You know, sucking them up.
Yeah, no.
Quite amazing. Yeah.
Behind electrostatic spore traps, I just don't think they can cope with that much volume of air as a Burkhardt spore trap or a rotarod spore sampler. So yeah, and the less air you sample, the less likely you're going to find fungi.
And I mean, there's not much airflow at all, really, in the glasshouse environment.
Yes, it does make it very difficult.
And are the spores sticky, or
sticky in what way?
Do they release very easily from the conidiaphore, or not? Does it require a good waft of sort of air to move them, or will they drop easily?
The stronger the air, the better.
The better.
However, they do release at around what was it? Trying to think back at one of my experiments. I think it's somewhere around definitely 0.5. I think 0.2 meters per second, very easily released. In terms of speed, it also depends how long the conidiophores have been growing spores. Because if, for example, in my image, you saw 3 spores attached to a conidiophore, that was because it wasn't shaken or mechanically disturbed. If there's a lot more disturbances or movement, usually spores will be attached, even if it's just one.
Okay.
Once it's ready.
Brilliant. Well, Anastasia, I think that's all the questions. Yes, that's all the questions. So thank you so much for your talk. It was really, really fascinating, very interesting. I really enjoyed it very much.
Thank you very much for having me.
So now we'll move on to our second speaker for the evening, Barsha Kalita. So if Barsha could join us now, if she can hear.
Hello.
Hi, Barsha. Hi. So let me just introduce you. So Barsha is currently pursuing a PhD in bioinformatics at the School of Life Sciences, Pondicherry University in India. She completed her graduation and post-graduation in zoology from Cotton University, Assam in India, in the years 2016 and 2018, respectively. She joined as a PhD student in 2018 and is currently writing up her thesis. Barsha's work focuses on an opportunistic fungal pathogen, Rhizopus delamere, causing mucormycosis. And as a biomathematician, she's utilised publicly available transcriptomics data of the pathogen, of its germinating spores, and identified novel non-coding transcripts with key cell wall-related enzymes that can be used as potential targets against mucormycosis, and also provided an insight into the onset of spore germination process through gene co-expression network analysis. So this all sounds absolutely fascinating. So it just leaves me to introduce the title. So the title of Barsha's talk this evening is Identifying Molecular Targets in Germinating Spores of the Mucormycosis mold Rhizopus Delamere. So over to you, Barsha. Thank you very much.
Thank you. Thank you. Let me just share my screen. So, are we good to go?
Yes, that looks good. Could you hold your microphone closer to your mouth?
Sure.
That's better.
Sure.
Thank you.
All right. Thank you, Ali, once again. So today I will be representing a part of my PhD work, which is entitled, again, Identification of Molecular Targets in Germinating Spores of Mucormycosis mole Rhizopus dilemma. So before we begin, let us just have a brief introduction on the on mucormycosis, which is an angio-invasive infectious disease caused by fungal pathogens belonging to the subphylum of Mucormycotina and order Mucorales. And among the different species, the species Rhizopus dilemma is the most common one, responsible for almost 70 to 80% of the mucormycosis cases, and it ranks the third most prevalent fungal disease after candidiasis and aspergillosis, with a high morbidity and mortality rates, and it usually establishes infection in immunocompromised patients. So it has got several manifestations, clinical manifestations, such as the rhinoorbital, cerebral affecting the brain, pulmonary affecting the lungs, cutaneous affecting skin, gastrointestinal, and various other forms. And the current treatment is based on a combination of antifungal medications like amphotericin, isoflurane, as well as the surgical treatment. But even after this, the incidence rate for mucormycosis still remains high, ranging from 0.005 to 1.7 cases per million populations, and the occurrence of mucormycosis is approximately 80 times higher in India, and it was significantly increased, especially after the COVID pandemic. So this gave us actually and when we compare it with the rest of the countries, developed countries, even in that, India ranked highest among them. So this actually gave us a scope to search more extensively the molecular mechanism that regulates the pathogenesis in mucorales. And then we came across a research article published by Septon Clark et al. In the year 2018, where they highlighted the role of germination to play a very crucial part for the disease progression. So the overall process of germination, which is actually the transformation of the dormant spores to the vegetative, or what we call as the infectious spores, happens through a number of phases, and where we can see some noticeable changes like the cell swelling, then water absorption, cell wall reorganization, polarization, hyphal tube growth, and emergence. So what actually basically happens once the dormant spores goes inside through the uh, through the nasal cavity inside the body of the host, especially those who are the immunocompromised. So when the appropriate conditions are met, the dormant spores germinate, and the disease actually begins with the invasive growth of the hyphae inside the blood vessels, causing tissue infection and necrosis. So with this concept, the aim of our study was to concentrate more on the germination process of these species, Rhizopus dilemma, in a more holistic way using weighted gene co-expression network analysis. And then, since the fungal cell wall plays very important role in fungal survival, so we also tried to, our second aim was to focus on the cell wall related enzymes, and with the emerging role of long non-coding RNAs, also known as the LNC RNAs, in response to transcription regulation, cell wall modeling, and nutrient response, etc. In many different fungal species, our aim was also to identify novel long non-coding RNAs or LNC RNAs in this species. So with this, we proceeded. So first, we took a data set, which is actually publicly available transcriptomic data set, like from the same author, Septon Clark et al. So it had 30 paired end samples that were sequenced on Illumina HiSeq 2000. So all the 30 samples were named based on their phases of germination. So for the 0 hour, it was termed as the resting phase. From 1 to 6 hours, it was named isotropic phase 1 to 6 respectively. Then for 12, 16, and the 24th hour, hyphal growth phase 1, 2, and 3 respectively. So all these samples were having 3 replicates, making it a total of 30 samples. Then, after downloading the FastQ files for these samples, they were quality checked using the FastQC tool. It is like a protocol we do. And once it is done, it has passed the quality check. So we proceed for alignment of those short reads of the samples using with the reference genome of the which is in our case is the Rhizopus dilemma. So after this, when we using the HiSet2 tool, we aligned it with the reference genome and found an overall of 82% alignment of those short reads that were uniquely mapped. And then after this, so we are like, it's like we are good to go. And then the short reads were after we used another tool called string tie tool. So for the quantification and also identification of the novel transcripts and of the samples. So I'm using the word transcript and not genome because our aim was for identification of LNC RNAs. So what are LNC RNAs? These are RNAs longer than 200 nucleotides that are not translated into any functional proteins and are involved in the regulation of cell wall differentiation and development in plants and animals. So based on the position of the LNC RNAs, so they have been classified into different types, and there is a tool called GFF Compare. They have given the class codes based on their positions, such as U, which stands for the LNC RNAs that are present in between 2 genes. Then intronic is for the LNC RNAs that are present between 2 exons. Antisense are those LNC RNAs that runs in anti-parallel direction of a gene. Then O represents other exon overlap with the reference transcript, and E is the single exon transcript overlapping reference exon. So using the tool, we identified a total of 6,571 transcripts that were based on their number of transcripts were classified as follows. And among them, we could see that highest number were present for intergenic, followed by intronic, antisense, and very few for O and none for E. So after being classified as non-coding transcript, we then tried to see the coding potentials of these transcripts because we did not want any of them to be having any coding potentials. So the tools we used were LNC RNA, field LNC and PLEC. So through these, we screened out those transcripts that were having coding potentials, and from then we took those that were not having any coding potentials that came to around 4,800 transcripts. And to make our study more robust, we again went for BLAST using different databases, TREMBL, SPROT, PFEM, and further NCBI non-related database for BLAST to go. And finally, with the total number of novel LNC RNAs that we got were 2,569, among which most of them were intergenic, representing U, 218 were antisense, and 5 were intronic. Now, moving on to weighted gene co-expression network analysis. So before we begin, we did a PCA or principal component analysis for our samples, and this is a 2D PCA plot where we can see 41% variance in PC1 and 19% variance in PC2. And this shows a clear demarcation of all the 3 phases of germination. This is hyphal for red, resting for blue, and green for isotrophic. And what we found that since we can proceed further, since the clear demarcation was met, we can proceed further for WGCNA analysis. So As we know, most of the biological phenomena follow a scale-free topology. That means there are some genes that plays a majority role for all the downstream processes, and these are called as the hub genes. So now, uh, for WGCNA, as for a scale-free topology, we choose the lowest possible power term, or beta, for a scale-free network. So here, in our case, to proceed with WGCNA, we chose our lowest power beta, having a correlation coefficient greater than 0.8, and proceeded for WGCNA. And then we, after this, we do hierarchical clustering. What it means that the set of genes, or we call it as modules, having high expression similarity, will be clustered together based on the hierarchical clustering, and further merging of those modules, and with a dendrogram cutoff of 0.2, we finally got a total of 40 modules. So these 40 modules are represented here, so these are the set of genes or transcripts having high expression similarities represented on the y-axis, having their unique colors they were assigned their unique colors. And in this correlation heat map, what we do, we try to link or associate these modules with some external trait. So here in this case, our traits are the different phases of germination. R represents here the resting phase, I represents isotropic, and hyphal for H. So this blue to red color here represents negative to positive correlation. So we are trying to correlate the phases of germination with the modules. And what we found here was a particular module, which is the purple module, to have a very was significantly, as we can see, significantly highly expressed only in the resting phase, having a highest correlation value of 0.95. So this indicates this module to be our module of interest, as it is expressed only in the resting phase. So this also corroborates with the expression heat map of the purple module, where we can see that the purple module has a distinct pattern of expression only in the resting phase. So we proceeded further, taking this phase to be this module to be important, as it is expressed only the initial phase of germination, like the resting phase or the dormant phase, indicating some importance of this phase. So after this, we proceeded with gene ontology annotation of this module and we found the 1607 accessions to have gene ontology information. And if we can see here in the graph, they were having mostly molecular functions and biological processes, and the biological processes that were mostly enriched were protein phosphorylation, carbohydrate derivative metabolic process, phosphorylation, cellular response to stimulus, and the molecular functions that were mainly involved were hydrolase activity, binding activity like DNA binding transcription factor, protein binding activity, then transferase activity, phosphorus containing groups, etc. So after this, the next part of our work was to focus more on cell wall related enzymes. So as we know, carbohydrate active enzymes or enzymes are those groups of enzymes involved in polysaccharide synthesis, degradation, and modification. And based on their activities, they are classified into different modules, which is glycosyl hydrolase or GH, auxiliary activity, glycosyl transferase, polysaccharide lyase, carbohydrate esterase, carbohydrate binding modules. And in this purple module, we were able to annotate 63 enzymes using a tool, DBCAN2 tool. So in the enzyme module distribution, the highest module that was present was GT, or the glycosyl transferase, followed by GH, AA, PL, CE, and CBM. And if we look at the frequency of enzyme families in each modules, so in case of the glycosyl transferase, GT2 that encodes for chitin synthase, GT15 manosyl transferase were found in abundance, meaning that these enzymes are very crucial. during the initial phase. Then in case of GH, polygalacturonase, chitin beta gluconyl transferase, chitinase were found in abundance. And for AA and PL, lactase and glucuronidase were found to be in abundance. Moving on to the network analysis. So this is the overall pictorial representation of the gene co-expression network, where we can there were 264 nodes connected to 11,817 edges. So where we can see the 102 accessions were having gene ontology information, or they were coding transcripts, and there were also 60 that were represented here in the purple color, and there are 65 predicted LNC RNAs and 6 caseins represented in yellow and red colors respectively. Further, the networks was functional enrichment of the network revealed binding activities, majorly zinc ion transport, substrate specific transporter activity, majorly solid protein antiporter, hydrolase activity like carbohydrate substrates. Then we went for further subclustering of the network, and we found two main subclusters. Subcluster 1 that had mostly 4 caseins, 41 LNC RNAs, and 112 protein coding accessions. And among those 4 caseins, those were mainly again GH15, 28, GH46, and GH47. Then in subcluster 2, we found 1 casein to be co-expressed, and this was again GH16 and 3 LNC RNAs. Then we also tried to see the interaction of those LNC RNAs that we found present in the network, and the interaction between the LNC RNAs and the mRNAs in the network. So in the subcluster 1, among the top 11 LNC RNAs, except for 1 LNC RNA, almost all the LNC RNAs were having interaction with the mRNA. And in subcluster 2, all the 3 LNC RNAs were found to be interacting with the mRNAs in the main network. Likewise, even we checked for the caseins in this network. So we found 4 important caseins to be having interaction with the LNC RNAs. These were again chitin GH16, which is chitin beta 1,6 gluconyl transferase, GH28 that codes for polygalacturonase, GH47 alpha manosidase, and GH15 alpha manosyl transferase. So the whole germination process was explored through our study in a more holistic way using weighted gene co-expression network. And through this study, we were able to identify novel LNC RNAs, mostly which were intergenic and antisense. And the co-expression network revealed important molecular functions like the binding activity, substrate-specific transport activity, and hydrolase activity. Then cell wall related enzymes like chitin beta gluconyl transferase that encoded by GH16, GH47, GT15, and GTH28 were identified. And among this, the casein GH16 is actually and can be said to be a very important target because it doesn't have any human homologue. And actually, I have in the rest part of my PhD work, I have actually used this enzyme, taking it for my further studies, and it actually shows very good results. And co-expression as well as interaction studies of LNC RNA, mRNA was revealed that transregulation of some crucial enzymes. So this work has been published recently in the Mycology Journal. This is the research article with its DOI. And thank you so much for listening to so patiently. Thank you.
Thank you very much indeed, Barsha. That was great. Gosh, fascinating. Really very interesting. I'm going to just see if there are any questions for you in the chat, first of all. Okay, let's see. Okay, there's a question here from Alan Clark to everyone. As it's much easier to detect we have to be careful with this. Tribal types in India than Western ethnicities. Have you spotted any genetic signals in different populations, and what are they?
Okay, so in the current part of my work, I have not uh, gone through that or have not performed that. Actually, for this kind of work, we'll be needing data. We, being a bioinformatician, we usually go or try for publicly available data. So whatever data set is available, based on that, we usually perform our work. So currently, I have not performed and we have not gone through that. So, yes, this is actually an interesting question. It will be very interesting to study as well for future. Yes.
Mm-hmm. Great. So it's really interesting that the disease sort of manifested itself as a real serious problem in India following the COVID pandemic. Yes. I'm just intrigued. Why do you think this plant pathogenic fungus emerged as a serious human pathogen? What were the drivers for this change, do you think? Or is it a simple case of sort of just being opportunistic?
Yeah, that's a very intriguing question, actually. So maybe during the course of evolution, something might have happened. We need to explore more, but I would like to add an information that the genome of this particular strain has it has undergone like a whole genome duplication. So, and there could be possibilities for that has increased the pathogenicity level at some point, maybe. So yes, that is.
No, that could well be it, actually. Yeah.
Yeah.
So another question here. What is the cause for India to have such high rates of mucormycosis?
Okay, so if we talk about mucormycosis in general, as I already said, it usually affects people who are immunocompromised. Also, for example, people having more diabetes. So in India, the rate for diabetes is very high. People having the rates of diabetes is very high. So one of the main reasons is for that. And if we talk about COVID, yeah, during incidents of COVID, where especially the second wave was too high in India, and with the conditions of diabetes mellitus, then the COVID, the whole scenario, and the conditions in the hospitals all taken together, these are some of the reasons for this disease to such a high rate in India.
I mean, it's quite interesting that you mentioned sort of diabetes there, because I'm guessing, I mean, when I think about, spores and spore germination, I mean,
Yes,
yes. Nutrient levels are really
yes.
important, so
yes.
particularly glucose.
Yes.
So maybe that, there's something
Glucose,
in that.
yes, and diabetes mellitus, and there's also the one related to iron. Free irons usually get available, yeah. So
okay.
Yeah, so that is one of the reasons.
Great. So another question in, how does this help us to know how best to treat infection? I've heard that a strong gut microbiota are important to suppress it. So is the gut microbiome, does that play a role, do you think?
Can you just repeat it once again for me, so I can...
Yes, yeah. So the question was sort of, how does this help us to know how best to treat the infection? So your data, so what you've done so far, can you come up with any solutions or any okay. direction as far as treatments maybe for mucormycosis? Yeah.
Okay. So like from what we concluded, we found out some of the important, especially enzymes or cell wall related enzymes. So those can actually be targeted. So if we talk about the enzyme GH16, So chitin gluconosyl transferase. So like this fungal cell wall is made up of the chitin and the glucan layer. So when they, this enzyme helps in the like cross-linking of this, so of the cell wall. So when this happens, so this actually means that this enzyme is actually very crucial for the fungal survival. And since no human homologue is present, so if we target that enzyme, so it actually can in future maybe can be used as a like a target for the drug, for any drug. So yes, we can say we have found few targets, and among them the important one being GH16. Yeah.
Excellent, good. So another question, Barsha, could you hold your microphone up to your, just so we can hear you? No, it's interesting. We don't want to miss anything here. So another question for you. So someone said here, so fascinating talk. Thank you. Is it possible that climate change has played a part in increased infections?
Maybe, yes, but without data, I cannot assure not say like anything currently. But yes, given the situations now, maybe it can maybe play a part. But since it is without any like evidence or factual evidence, it's not possible for me to currently say it in like with more confidence. I cannot say it currently.
I have a question for you. I mean, it's more about the biology, really, just trying to, I'm sort of fascinated by some of the early slides. So mature resting spores are dehydrated, and then they sort of sense environmental cues. That could be glucose, it could be all sorts of things, but it involves sort of water uptake. Were any of your targets aquaporins? So these channels that allow small and uncharged molecules to enter. I mean, that's sort of a prerequisite for sort of germination, really.
Yes.
It's and we think it's probably now a regulated process. So it's not, water just doesn't go in. It is regulated through sort of cell membrane bound proteins like aquaporins. And I just wondered if any of your signals were for aquaporins in Rhizopus in those early in the eye sections of your analysis.
Yeah, it's like a very, very good question. So we talk currently I cannot recall if exactly aquaporin were there, but maybe like I was already mentioned, transporter activities were enriched in the network. So if I go deep and search exactly what were they, so yes, we did find the transporter activities. So maybe there are like maximum 90% possibility for aquaporins.
In your purple module, you certainly had binding activities going on there.
Yes.
So another question is, so when these sort of resting spores they evade host defenses getting in through the lung cavities, so and enter the macrophages, do they lie dormant there, and are they dehydrated in that stage?
So actually this question, like the mechanism, maybe I have to like go through more a bit for this question, because I did not go like full into the mechanism.
Right.
Yeah, so
no,
currently
I'm just interested in the sort of biology.
Yes.
No, don't worry.
So being a bioinformatician, I actually need to go through more maybe for the mechanism. I do have to check a little bit more about it.
It's just, I mean, it's obviously very interesting. And there's another question here is, so mucormycosis, do you think it could spread to the rest of the world?
This has actually spread to most of the parts of the world. So yes, it has spread. So actually, this Rhizopus dilemma, it is actually a plant pathogen.
Yes.
So yes, so
I know
it
it
is
well as a
present.
plant pathogen, but
Yes,
not.
yes, yeah. So it's very interesting actually. So to how it, that is actually a topic, how it evolved from a plant to a human pathogen. So that is also very interesting. So and this if we can find it almost, this spores at least we can find it almost everywhere. It's very common
Oh,
actually.
it is.
So
Yeah.
it once it enters into a compromised patients, so that is when actually the disease happens. So it depends on the places and everything, and the appropriate conditions where it is germinating also.
Yeah, because I mean, in nature, I mean, I'm more of a plant pathologist than
Yes. Yeah.
so in nature you see this. I mean, Rhizopus, it sort of it likes sort of, you know, fleshy vegetables, and
Yes.
you know, and the pH is normally about, I don't know, 4 5, something like that, and that sort of seems to be quite important in terms of triggering germination and so on, and
Yes.
and I think these aquaporins, you know, do play a role
Maybe.
in that
Definitely.
in nature as a plant pathogen. So it'll be interesting to see how that equates in the human model, and
Yes.
what the environmental cues are, you know,
Actually.
in the body. Yeah.
Yes, actually, and this is actually like a preliminary work, so we
Yeah.
can investigate more further. So since this is now like open to the public, so people can explore more on this. So yes.
Great, thank you. And one more question here. As your investigation seems to concentrate on the germination period, is the chitin enzyme mainly present to deal with sporopolline, or is it engaged in the infection?
Or polline, this word actually I'm
Is
not
Yeah.
familiar with. So
So Alan, if you're still there, if you could, I don't know whether you can enlighten us with sporopolline. Yeah. Okay, so it's about, so okay, breaking out of the spore.
So in so the what we found in the module was the enzyme that was mostly abundant was chitin synthase. So this enzyme, we all know it have it's related to more the synthesis of chitin, and chitin, as we know, plays a very important part in the cell wall. So yeah, we found even there was also chitin, I'm forgetting. So yeah, which cell wall remodeling happens. So sca- so so those enzymes were also involved. Yeah.
Fantastic, great. Well, thank you very much, Barsha.
Thank you.
There aren't any more questions that I can see in the chat, but that was really a very, very interesting talk. You know, very, very important and relevant topic just at the moment. So, thank you very much for that.
Thank you. Thank you so much.
Thank you. If we can have Anastasia back on the screen as well, that would be great. So it just leaves me to thank everyone for joining us this evening for these fantastic talks. I've enjoyed both of them. They've been really very informative. And a big thank you to both of the speakers. They're very, very clear indeed, and very, very interesting. And I think it just leaves me to wish everyone a very happy festive season, And thank you very much for joining us this evening. And I'll hand over to Sally now to close things off for the evening. Thank you.